DNA, the building blocks of life. Today, we're going to be talking about DNA replication, how and why it's important. What is DNA? DNA, or deoxyribonucleic acid, is a double-stranded genetic material made of nucleotides that have a sugar-phosphate backbone and nitrogenous base. DNA coils around histones many times, forming chromosomes. I have made a 2D model on Google Slides to represent DNA replication. But before we get to my model, let's talk about the most famous DNA model, the Watson and Crick model. In 1953, James Watson and Francis Crick proposed a new DNA model still widely used today. Guided by ideas from other scientists like Linus Pauling, Phoebus Leverine, and Erwin Chargaff, the Watson and Crick model accurately depicts DNA as anti-parallel, double helix, double-stranded and complementary base pairings, and with nitrogenous base edges remaining exposed for protein access. So now let's talk about the DNA structure. DNA has three main parts. The first is nucleotide pairing. This shows the primary structure of DNA, sugar, phosphate, and nitrogenous bases, where phosphate is attached to a 5' of a pentose sugar, and it has a nitrogenous base of either G, C, A, or T. The nitrogenous base is connected to the 1' of the sugar. The next one is the sugar-phosphate backbone. The phosphate from a nucleotide attaches to another nucleotide through the 3' of a sugar. So basically, there's a nucleotide pairing here, and the phosphate is connected to the 5'. The phosphate would also connect to the 3' of another sugar, and it eventually repeats until it becomes a big backbone that connects the nitrogenous bases. DNA is also anti-parallel, so each strand of nucleotide is opposite of each other. Finally, there's the complementary base pairing rule, where I'm going to butcher these names, adenine will always pair with guanine through 2 hydrogen bonds, and cytosine will always pair with thymine through 3 hydrogen bonds. Now let's get to DNA replication, and more importantly, or more specifically, DNA replication in eukaryotic cells. In a eukaryotic cell, DNA replication happens inside of the nucleus during the S phase of interphase. During this time, new cells need to be identical for daughter cells to copy and create new cells. Now let's talk about the importance of DNA replication. DNA stores genetic material needed to build and maintain an organism. And so we need DNA replication to help build, repair, and maintain our body, as well as copying the exact DNA of an organism to ensure future daughter cells have correct instructions to grow and repair correctly. Now let's talk about the DNA model. Here we have the Watson and Crick model, and if we were to unwind it, it would look almost like a railroad track. And so now let's go more in depth. Here we have the key with all of the different nitrogenous bases and enzymes in DNA replication. This is the DNA structure unwinded, and this is a helicase. The role of the helicase is to unwind the DNA, and it basically unzips the double helix. The place where DNA replication starts is called the origin. Once it opens up the DNA, the helicase creates what's known as a replication fork, which is where DNA replication happens, and the helicase will always go in one direction. Next, because we don't want the DNA to coil back and become DNA again, single-stranded binding proteins are deployed. They attach onto DNA strands and prevent the strands from coiling back and coming back together. And because unzipping DNA causes strain, topoisomerase is deployed in front of the helicase to prevent supercoiling and damage to DNA structure, untwisting DNA when required. Next, polymerase III needs to come in and build new nucleotides. However, they need a place to start. Primase finds a place for polymerase III and primes them to work. They deploy RNA primers, which are around 5 to 10 nucleotides long, on both strands. This helps polymerase start building nucleotides and continues the chain started by primase. Polymerase III can only build in the 3 to 5 direction, and so on this strand, since it goes 3 to 5, it's just following the helicase. It becomes a leading strand. However, the one on the bottom becomes a lagging strand because it goes from 5 to 3, and it has to keep up with the unwinding helicase, leading to fragments and gaps between each section, which is called an Okazaki fragment. Eventually, the RNA primer gets replaced by DNA nucleotides, since the primer is RNA and it's not meant to be RNA. then they're converted by polymerase 1, and boom. Finally, to connect the Okazaki fragments, ligase comes in and acts like glue, making sure all nucleotides are joined together. And if we zoom out, and zoom out further, and zoom out even further, you can see that there's multiple replication forks. As two replication forks are formed at the origin, replication bubbles are formed. During DNA replication, the process needs to be quick, so multiple replication bubbles and multiple forks are formed so that DNA replication happens quicker. It's almost like pulling something apart down the middle. You rip it apart, and then it basically becomes two. Just like him, he's pulling the doors apart. Now, some final notes. The two DNA strands are called semi-conservative, as both DNA strands have one original strand and one copy. Polymerase can also proofread the inputs, rarely making mistakes, and if it does, polymerase can go back and correct these mistakes. DNA replication is vital to species continuation. Through DNA replication, genetic information is able to pass on to offspring correctly. It allows the daughter cell to get identical genes from the parents, helping grow, repair tissue, and allow normal development. It also makes sure correct genetic material from the parents is actually passed down to the offspring, because if it isn't, mutations could occur, which could cause a fall in survival and reproduction. Now, some benefits of using Google Slides is that Google Slides in 2D models makes it easier to understand and simplify the process. And if you have enough slide experience, you can make really wonderful animations, like unwinding of DNA or actual moving enzymes. Also, making a color-coded key is easy and makes enzyme identification easier. However, a lack of scale on the digital plane means we can't accurately compare the size of what DNA replication truly is, and simplification can cause oversimplification, making the process potentially unclear. Good animation on Google Slides is also really difficult, and you need a lot of experience to do so, and different interpretations can also become possible on a 2D plane. Alright, well, that's everything. Thank you for watching.